Today, what we're going to do is talk about the papers on sort of ecological, sort of the two classic papers on using ribosomal RNA analysis to do ecology. And there's this one on UniFrac, which is now basically the tool used to do ecological assays, comparisons of ribosomal RNA data. And there are both sort of scientific reasons that it's become the tool and also just technical reasons. So the people who made the most widely used software that people use for ribosomal RNA analysis came from Rob Knight's lab, and so they embed their tools inside their package. But it actually is a really fast and powerful way to do beta diversity calculations, basically, for microbial communities. And so what I'm hoping to do here is to get you guys to help discuss the different parts of UniFrac. And basically, what they do in their paper is they, as most, many scientific papers, especially papers on new methods do, is they start off their paper with a complaint or two about how awful things are done by other people. Sometimes people are polite about it, sometimes they're not. So do you know what the main complaint was in this?
You're saying that they don't really take into account sort of the differences, like abundances. There's a lot of sort of, I think I might be getting that paper.
Yeah.
So they basically was saying if something, so we talked about creating that cutoff for identity,
Mm-hmm.
and if it doesn't fall within that cutoff, that 2 or 3%, then it all gets grouped the same way. That has the same unrelatedness to that cutoff.
Right. So, and that's bad because
then you can't build it doesn't allow you to kind of construct any sort of relationship.
And it doesn't count all the information. Basically, you're throwing away data. So what they go through is basically this argument that there are methods to do this. They weren't saying that this is the first time anybody thought of doing beta diversity comparisons from microbial data, but they don't account for the different degrees of sequence similarity that you would see between data if all you're doing is collapsing things into OTUs and then counting OTUs. So this is what I was talking about last time. This is the list-based approach that many people used and still use to comparing communities, where you make a list of species that are present, or genera that are present, or families that are present, and then you compare the presence and absence of those across environments. And that doesn't take into account the relatedness. And so, they go into a specific complaint, which is this percent identity cutoff. But, people have made this complaint before when, you were comparing plant species, presence and absence. The plants weren't determined by any percent identity cutoff. They were taxonomists who went in and said, this is species 1, this is species 2, here's the list. That doesn't take into account whether or not species 1 and 2 are in the same genus or different phyla. And this is the same issue. And then here's the start of the feud between Rob Knight's lab and Pat Schloss's lab, the other big lab that develops beta diversity tools for microbial data, which is they say, here are some examples of methods that suck, that don't do this. And ours is just going to be so much better than these old school, boring, useless methods. And as far as I can tell, they've never talked to each other since. But I was at a meeting once where they talked back to back, and it was the closest I've seen to a fist fight happening at a scientific meeting. So then they say, how do you fix this, right? So how do they fix it? You include the similarities of what?
The two organisms. It's based on the idea that like two communities that have been separated by an environment have diverged from each other. And so if you count up the shared or not shared, the unique sort of tree length that, or branch length that is associated with each sort of community or group of organisms in an area, then you can sort of jump over the problem of having things that are close together because it's included in the analysis to the degree.
Right. So they basically make the argument that the way to fix this is phylogeny. So rather than using a straight cutoff that says yes or no, you are in a group, species, genus, family, OTU, whatever that is, that you could use phylogenetic distance as part of your metric. And they did not invent this. So this has been widely discussed in particular in plant and animal communities for a very long time. The place where it, I think, gained the most traction was in conservation biology, where people were asking questions, and they've been asking this for a long time, but I think it started really to be more mathematical in like the 1970s and 80s, saying I have 7 parks or 7 patches of land and I want to protect some of them. How do I choose if I can't protect them all? How do I choose which one to protect or which ones to protect? And historically, when people did this, they would list the endangered or threatened species in those environments and count the number that were in those places. And then people came along and said, that's not quite right. If you want to preserve genetic diversity, you need to weight by the distance organisms are from each other, and at least include that as a component. So in the conservation field, there was a huge amount of development of these phylogenetic-based tools for comparing community structure and weighting the distances between them and actually using it for a very applied purpose. And so they then go through a few of these. So they say, how does one use phylogeny? And I don't know if people are familiar with these, but there's a whole suite of these methods. Some that came from people trying to do conservation biology, some that came from other methods. So there's this p-test and this FST test, and these are the ones that people had been using with sequence data for doing these types of phylogenetic comparisons. And they complain again about these. So do you remember what the complaint is about these? So here they're saying people should take into account phylogeny. We're not the only people to have thought of this. Here are all these other phylogenetic-based methods.
I think it says that it only sort of tests whether or not differences are significant. It doesn't really say anything else about the differences between multiple communities.
Right. So they do make complaints about how to do statistical analyses using some of these other methods, and they argue that they have a better way with Unifrac. But they make an earlier complaint.
Are they saying it says neither measure accounts for branch length information?
Exactly.
So is that referring to evolutionary rates?
Yeah. So it's basically the metrics that were used. The way they worked was not taking into account all the information in a phylogenetic tree. And what they argue is that if you have a phylogeny and if you have information on branch length in that phylogeny, then you should use it. And it's not just different rates of evolution. So there are basically 2 different ways that people draw and use phylogenetic trees. The easiest way is what's called a cladogram. In a cladogram, the only piece of information in the tree is the branching order among organisms. And so you could have, let's draw it frequently. Something like this. And you know that what this tells you is that A and B are sister taxa. They share a common ancestor with each other to the exclusion of everything else. A, B, and C are another group. D and E are a group, and F is the outgroup relative to everything else. But they don't tell you anything about the time or the length of separation of any of these organisms. And it doesn't necessarily have to be anything about really evolutionary rates per se, because what this doesn't tell you here is where on this branch, after this ancestor happened, before this happened, did C branch off? Did it branch off really close to A and B? Or did it branch off just after this other branching point? So that's independent of rates of evolution. And that's not captured at all in a cladogram. The cladogram just shows the order. And a phylogram, by adding in branch length, captures unequal rates of evolution and the timing of when branch events happen. And by doing that, and by developing a mathematical method that can allow you to use a phylogram instead of just a cladogram, because a cladogram is much easier to use, they're arguing that this should be superior because there's more. Again, it's basically an information, you're throwing away information if you don't do that. Does that make sense? So that's basically their introduction, and this is really common in methods papers. Their introduction is, here's what everybody is doing, or what most people are doing, and it's fundamentally wrong. And then some people have figured out that it's fundamentally wrong, but we can do better. And that's sort of a really common strategy. And so then they get into basically what they propose to do as an alternative, as the way to incorporate phylogeny and branch length. And that's where unifrac comes in. So does anybody want to try to explain verbally or mathematically what unifrac does? Well, they didn't do a very good job in this paper of explaining it, unfortunately.
My understanding is that you have a bunch of sequences, and you have created a tree of organisms from those sequences. And then for organisms that you label those organisms based on basically where they've come from, like what sample they've come from. And then you take the branch length that is unique to one sample. So like if A and B were from two different environments, then the branch length that would be unique to B would only be after that last node. Whereas if, let's see, if A, B, and C all came from the same environment, then you would include all of the nodes branching off from, down where, go back. So one node back, two nodes back, three nodes back. No, next node forward. Yes, then you would highlight all of the unique branching node going to A, B, and C, because that is unique to that environment.
If they're from the same environment.
Yeah, sorry. Yeah, all of them are from the same environment. I changed.
Yeah.
So then all of that branch length, so there would be more unique branch length in that case than the first phase. And then you can create a distance matrix between the communities. So say, one community has more unique diversity.
All right, so to calculate that distance matrix, what's the formula? Which they don't show you, but
yeah.
you have to figure it out by reading between the lines over and over again.
Unique fraction, right?
Yeah, so what is that?
It's just the fraction divided by the total.
Right, so it's the unique part
Includes in the name.
Divided by,
over the total, but how do you calculate? So let's say you're comparing a environment 1 and 2, and how do you calculate? What do you do to calculate the unique part? The total branch length's easy, right? That's the total branch length of everything in the tree.
So it's looking at the sort of cumulative branch lengths that are, I guess, highlighted in gray that they were talking about.
Right, but so let's do it for environment 1.
Yeah.
Here's the unique part for environment 1, and then there's another unique part going up to E. Is that, we sum up that?
And then F is well.
And then what?
F also.
Oh, right, so, and F.
Yeah, but you wouldn't include the nodes, but you wouldn't include
I didn't, yeah,
the
so this
bit
is,
going up to, yeah.
and then,
That's
are we
what it
done?
looked like to me, like you would do that, but maybe I'm missing a normalization step.
There's no normalization, but are we done? We've left out the other half,
We have
which is
to-
you also calculate the part that's unique to environment 2. And now you sum them. So you're summing up the total that is unique to either
Oh,
of your environments out of the total tree length. And this took me forever to figure out because they don't really make it completely easy. They just thought, you know, we're just going to say the unique portion and it's going to be obvious to everybody. So it's this. The sum length of all the unique branches going up to either of the two communities divided by the sum total of all the branch length. So it's a measure of the uniqueness between two communities, right? So it's not just the uniqueness of one, but it's this uniqueness between two. And they go into then a lot of text when they're writing up about the method. So they did this really weird structure in the paper, and I still don't totally understand. So they have the sort of introduction and some questions that they propose that this can be answered to, and a description of Unifrac all in the introduction of the paper. And that's a little strange because Unifrac is a new thing. It should be in either the methods or the results, or, I mean, it doesn't, that description doesn't really belong in the introduction. But anyway, justifying it sort of makes sense in the introduction. So they go through, what we just sort of went through, and they go through some of what you were pointing out, which is that you can use, by doing this, it's fast, and you can do statistics on it, basically, by a variety of methods. So it's fast. Unifrac can compare many samples simultaneously. Some of the other methods that people had used before, even ones that didn't take into account branch length, were slow. And the more important thing is it incorporates this information about the different degrees of similarity. Well, we just went through this, but they sort of try to explain how it calculates in text and in figure. I think the figure is very helpful generally, but again, it doesn't explicitly tell you in the figure what the formula is for calculating the Unifrac distance. So if we just go through the figure, all that other text is just basically re-explaining the figure. So why did they show you these two trees here? What's the difference between these two trees? The squares and circles correspond to different environments. Yeah. Right, so they're inventing two extreme cases to highlight what Unifrac is doing. So in A, the taxa from the different environments have close relatives in the other environment, and in B, the taxa in each environment have only close relatives within the environment. And unlikely to see a clean pattern like this in most environmental samples, but this is simply a demonstration of the method. And again, they don't show you exactly how to do this, but they talk about how you can take this data and do, quote, randomizations, which they don't completely show you how to do, and some people think they do incorrectly, but let's just pretend like they do it correctly. And they say they randomize the environment was assigned to each sequence in the tree. And that's what this basically is showing you in part C, is R is replicate, I think. And so they take, let's just say this was your tree and these were your environments. And then they take that same structure of the tree and they shuffle around the environments that would be assigned to the different branches of the tree. So here's, shuffling around the circles and squares, shuffling them around, and calculating unifrac distances of the replicates to each other. So this gives you, for that particular tree, that structure of a tree, a measure of just randomly, if the environments were arranged in some other way than what you see, what is the distance that you would see on average between any two sets? So this is an attempt to create a statistic that you could then use to calculate the significance of an observation of an actual distance that you observe. And there are many different ways you could resample data like this. And we won't get into it, but the problem for them comes later when they try and represent. So this is the simplest unifrac. And we'll get to the end of the paper where they talk about how to make it, in theory, more complicated. It doesn't take into account different relative abundance of taxa. You're just doing presence and absence and not relative abundance. And when they started to work on relative abundance, they had a very strange way of doing the randomization that, not a very strange way, a very simple way to do the randomization, actually, and it's not clear if it was the best way to do it.
So all this normal curve is showing is that there are more tree combinations possible when the communities are more mixed versus when they become, I guess I'm trying to interpret. So there are going to be fewer trees possible the more unique the branches get from.
No, so this is, it's very, so what they do.
It's too subtle for what I'm trying to.
No, it took me a year to figure this out, or more. So you have your environments, and in their diagram here, everything is unique to one, and every tip goes to something unique. They're not showing, things that are shared between both of them. So everything has, let's just say, environment 1 is plus, 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 minus, minus, minus. Environment 2 is minus, 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 plus, plus, plus. Those are so they went to environment 1, you sequenced 3 genes from environment 1, they show up in this part of the tree. You went to environment 2, you sequenced 3 genes, they show up in this part of the tree. And this is their fake, their example construct where you have close relatives within an environment and they're distant from the things in the other environment. And then they say, okay, for this, let's use this as our example. How can we calculate the probability, the random probability that we would observe something like this, given a tree that looks like this? So they basically say, the way they do this is they basically say, we have 6 taxa. And let's now, and 3 of them are from environment 1 and 3 of them are from environment 2. Let's now just randomly shuffle this and create new patterns where maybe it's and let's do another random reshuffling. So you haven't changed the tree at all. You're saying the tree is the same. But let's now say which environment those samples came is randomly shuffled. There are other ways they could have done this, but this is one statistical resampling effect that says, let's now just sort of randomize what environments map to the different parts of the tree and calculate now, compare this pattern, that's the real data, to these patterns. And what they do is they basically calculate distances. Well, no, they calculate, sorry, they don't even calculate distances here. Then they calculate that score, the unique over total, for this, pattern replicate 1. And this is the true data. And they calculate the unique, right? And then they do it again for replicate 2. So this is the unifrac distance for this resampling of the real data of 2 environments. And when you do that, you get a histogram of the distances that you observe with randomization of the data. That's what's plotted here. And then you can ask in a standard sort of statistical way, is mine an outlier on that normal random distribution? And what is the p-value that, can I calculate a p-value that would tell me what is the probability that this would have happened by chance? So they basically say you could do this for any data set that you have in any tree. You could take your real data set, resample it, create a histogram like this, or create an average and a mean and a standard deviation, and then calculate a p-value. Right. Does that make sense, what they're doing here?
Yeah, I was
misinterpreting
how they were using that graph.
Yeah, it took me forever. So, and that's basically what they, by doing this, and by making it relatively easy for people to calculate a measure of the statistical probability that what you observed is unusual, right? That it launched 1 million beta diversity studies of microbial diversity. And then they go into many other, things that you could do with the unifrac metric. And the main, so this is, statistical significance, number 1. And the main second thing is in figure D. So someone want to try and explain what this is? No.
It's comparing how different the communities are based on total phylogenetic distance of their community or of the organisms found in them.
Yeah.
And then it's creating a tree based on that.
Right. So it's a little So it is a little confusing what they're doing here. So they're doing this. They're calculating the real unifrac distance for this pattern. So you compare environment 1 and environment 2, and you calculate a distance. That's the unifrac distance. And then if you have a third environment, you now can calculate distances for all of them. So this would be distance from 1 to 2. And you can calculate a distance from 1 to 3 and a distance from 2 to 3, right? By just doing the same iteration with the other pairs of environmental samples.
And this is always just present, not present.
For now, it's just presence and absence of specific tips of the tree. In a paper a couple years later, they introduce how to weight it by the relative abundance of different taxa. And so you get a distance matrix. And there are many things you could do with a distance matrix, but in this paper, they cluster. This is what I was talking about in the last meeting. For this study, they cluster those distances. And now draw a cluster diagram, which is basically a tree. It's not an evolutionary tree. It's just a tree of similarity of objects. And so this, from their simple simulated model, the environment represented by the circle and the triangle are most similar to each other in terms of the unique fraction of branch length that they share. And you can see that very easily. Here they are on one half of the tree, nested together somewhat, whereas the other half of the tree is mostly represented by the squares. And so it's a pretty straightforward extension to say that environment with the circle and triangle are a little bit more similar to each other than either is to the environment with the square, right? Does that make sense? And so then they go back. That's it. That's unifrac. You can calculate a distance, and you can do statistics. Yeah.
What if the same taxon is present in two environments?
If it is present in two environments, so they didn't really show this, but they cancel each other. So if it was like that, then you can incorporate that into the same calculation, and now that's just a shared. This is included in the total branch length in the tree, but not the unique part of the tree. And they don't show that for simplicity, but that it is straightforward in the math. Does that make sense? It works the same way. So then they basically go into now their scientific questions they propose that this could be useful for. And this, again, was a little bit in their paper, just sort of multiple places. So they go through these in the introduction and then go through them again in the analysis and results. And they're not identical. I don't know if you noticed this in the introduction versus the results, but that's okay. So here's the first question, and we're going to do this pretty quickly so we can get to the other paper. How does culturing affect similarities between samples? And then they go through some text about this. We've already talked about this, that, there are many uncultured organisms out there in the environment, and they're basically asking now a higher order question, which is, when we culture things, are we getting a biased subset of organisms from environments, or is it just random subset of organisms from environments? And since you can calculate similarity and between communities and between samples and compare it to randomness, you could use their metric to do this analysis. And then the second question is, how cosmopolitan are bacterial lineages? That's basically how widespread across lots of environments. This sort of gets a little bit of your question. In essence, how many cases do we see where they're found across all environments, and can we use our metric to look at that? And they talk about the prior research on that area. And then they ask a very specific question, solely asked because that's the data that they had. Who would have come up with this as a question, right? You wouldn't get through your thesis qualifying proposal if you asked this without a reason. But that was sort of the samples that they were looking at, so they invented a question to go with them. And it's fine. At the time, there weren't a lot of data sets available, so it's a perfectly reasonable question. It's just, it comes from the data as opposed to comes from sort of theory. And then there's a reasonable amount of text on where this data came from and how they, mapped things to particular environmental samples and how many sequences are there for particular samples, and so on. There is some useful information in there, but I'm going to skip over it. This is, very useful and important, and I wish more people would do this to provide sort of a table and references and other information about data that they look at. This is, has latitude and longitude in here and whatever other metadata they could find and how many sequences. That's at least useful to have. They talk about, computationally, how did they do this? They used ARB, in part to get the data, and then they manually curated the alignments. They threw out anything that was not bacterial, since the studies were supposed to be going after bacteria, basically. And then, they go through basically the workflow that we talked about in the last class is what they did. So then they get into one other statistical approach, which is useful and important for these types of studies, which is something called jackknifing. Do people know what bootstrapping is? So when you're doing statistical studies, there are many ways of resampling your data. Probably dozens of ones, but two that are really commonly used are bootstrapping and jackknifing. Bootstrapping is probably more commonly used in these types of studies, mostly because someone wrote software again, probably for doing it. Bootstrapping is you take a data set, let's say you have 10 alignment columns in a sequence alignment, and you resample that data set and you create a new data set also with 10 alignment columns. But you resample the data set with what's called replacement, where you pull out column 7, and then you go back and resample the data set again, and you allow yourself to resample column 7 again. You could hit it randomly. So with your new data set, you might have column 7 represented twice, column 1 represented not at all, and all the other columns represented once. And this is what's called bootstrapping. And you take that new data set and you run it through all of your analyses, and you ask, how different is my answer when I analyze this new bootstrap data set than if I analyze my real data set? And you do it again and again and again, and you get an analysis of how much your result depends on the different portions of your data. And jackknifing is like that, but you don't do replacement. So if you had a sequence alignment with like 10 columns, you would just pull out 5 columns. And then you would do it again and pull out 5 other columns, but you never have any column represented multiple times.
So it's bootstrapping and jackknifing, seeing how much something biases your data to look a certain way.
Yeah, exactly.
Okay, so.
And it's used throughout all sorts of different fields as a statistical method. And basically, bootstrapping is resampling with replacement, and jackknifing is just resampling a portion of the data, and nothing gets represented multiple times, but many things get represented no times. And they chose jackknifing. I think it was easier to do in this case. Or I actually don't know why they chose jackknifing. And then they basically go through, first they talk about sort of significance metrics for their analyses. So first they ran some of these statistical tests and asked, just are the differences that we detect between samples statistically significant? Not what do they mean, but are they statistically significant? They show you what the compared samples were for SR1, S21, et cetera, and what the statistical significance and the p-values were. And then they use those distances to build a cluster diagram of the samples. Here's your cluster diagram of the samples, just like they did in that model. And they've added a little tag for what the samples represent. And then the thing that they didn't really talk about a lot in the methods, and they didn't show in their little figure 1 model, is they also do principal coordinate analysis, just like we talked about before, to now separate out the samples and look at whether or not the samples group in this principal coordinate space by any type of environment that they come from, basically, or any of the metadata. So if you go back to table 1, so any of the information that we have about samples map well onto this principal coordinate plot. So you should first ignore the information here, and they basically show, so again, you have this data with however many samples these are. You take the distances between each of them, and you try and find a way of representing those distances by collapsing it into 2 dimensions. And then they tried to come up with, does any of this make sense? So for this sample, they said, oh, look, these are all the uncultured ice and cultured ice water and sediment samples. And these ones are all the sediment and terrestrial impacted seawater samples. And these are the oligotrophic subtropical seawater samples. They're trying to make biological sense out of the similarity between communities. And I don't think it's worth going into all the details of this, but that's what everybody is now doing with every microbial community-based study, is calculating some, global similarity between samples and then trying to make sense out of it by looking at how metadata maps onto this. And they, have pages and pages and pages of discussion of what they think all of this means. But does that make sense, basically, what they're doing here? With this sort of principal coordinate analysis and then looking for how samples map onto the, yeah.
Can you explain the U over T again? Because what's the unique branch length?
So the U over T, let's just start from the beginning. We take sequences. Let's say we get 5 from environment 1. And we get 5 from environment 2. And 5 from environment 3. That make sense? Mm-hmm. We make an alignment of all of them. And we build an evolutionary tree of all of them. Make it incredibly simple like they did in their model. And let's just say, now you say, we have a tree that represents all 15 of the sequences. And now let's add a little bit of information onto this tree, which is just what sample did this thing that is at this point in the tree come in from environment 1, 2, or 3? Mm-hmm. And let's just say those all came from environment 3. And let's say those came from environment 2, those came from environment 1, these came from environment 1, and those came from environment 2. And now we want to calculate the distance from 1 to 2, and the distance from 1 to 3, and the distance from 2 to 3. These are the unifrac distances of environment 1 to environment 2, and environment 1 to environment 3, and environment 2 to environment 3. Does that make sense so far? So if we do distance from 1 to 3, which is the easiest, 1 or 2 to 3 here, we ask of all the tree here that is shared, of all the tree that maps to where we see samples from 1 and 3. So that's our total, is the branch length leading up to either 1 or 3. We ignore 2 here. 2 is not part of our calculation. So any part of the tree that goes up just to environment 2, we don't count in this total distance. So here's the branches going to 1. We count that total length of all the branches that we have to trace to lead up to the samples from environment 1. And we also count the total length of branches that we have to trace to lead up to all the samples from environment 3. Okay?
Yeah.
And that's, for 1 to 3, that's the total, the denominator. And now we calculate the sum, all the distance going up to branch 1 that is unique to branch 1 compared to branch 3, or environment 1 compared to environment 3. It turns out that that is all of the branch going up to 1. So that's going to be unique for 1 plus the unique going up to 3. Now it turns out that that's going to equal T. That's the whole tree is represented because I forced them to be separate. So that's going to equal 1.
So in cases when it's less than 1, that means you have overlap between the 2. Okay.
Yeah. And so this is a distance, and it's on a scale from 0 to 1.
Okay.
And so we could do it now for 1 to 2. Well, 2 to 3 is going to be the same because I forced them to be separate. And then we could do 1 to 2 is a little more interesting and complex. But does it make sense, basically, how you would do this now? And so that's what they do to get the data. They don't show you the distance matrix, but they fed that data into a clustering program to get this diagram. They're grouping environments by their distance. But the way to, I know the way to, in essence, think about this is that we now have distance from 1 to 2, distance from 1 to 3, and distance from 2 to 3, right? And you can think about the way to represent this would basically be in 3 dimensions, and you could force it to work such that you were in 3 dimensions, and you could force the spots to be in the right place such that the spots would be the right distance apart if you used 3 dimensions to represent three distances.
How do you feel about that?
That's the way you should represent the total data. The reason that you do clustering or principal coordinate analysis is if you have 20 spots, nobody can visualize a 20-dimensional, quote, hypervolume.
But just on a practical basis, how is that overcome?
That's what this is. On a practical basis, this is exactly how it is overcome. So everybody in all science takes data that has multi-dimensional space and collapses it into dimensions that humans can deal with. And so clustering is a way of collapsing it in a very specific way into 2 dimensions where you have a tree-like structure that tells you the order of similarity among the objects. That's actually best for historical data where there is a tree that represents the history of objects. But that's not what this is. So principal coordinate analysis is saying, I'm going to take this multi-dimensional space, in this case it's like 20 dimensions, and there's just no way for anybody to visualize that. And I'm going to run a mathematical algorithm called principal coordinate analysis in this case that collapses it into 2-dimensional space. And the positions that things are separated on 2-dimensional space is related to these coordinate formulas. So they're showing principal coordinate 1. There's a formula that represents that. They've collapsed the multi-dimensional space into this axis, now on the x-axis, via this formula that is represented by the principal coordinate 1. And then they collapsed the n-dimensional space into a second dimension that is represented by the y-axis. And so you're not seeing all the data here, but you are seeing a measure of how close environments are to each other by representing it on these mathematical formulas that are the x-axis and the y-axis. Does that sort? I mean, again, I don't understand the math behind this either, but I understand the point, which is to collapse these dimensions into places that humans can deal with. The way they showed it was to show PC1 and PC2 on one plot and PC3 and PC4 on another plot. They could have, done a 3-dimensional rendering of PC1, 2, and 3, or you could visualize this on a computer and look at, rotate PC1, 2, and 3, but, they chose, and I think it's probably better to do it in 2 dimensions, yeah.
So the space in between the objects is representing the relatedness of the environment?
Yeah, so the distance that objects are from each other here represents how far away from each other in that n-dimensional space that communities are. So if objects are, if this little spot here is more similar to this square in terms of its community composition than it is to this circle, which is much further away on that axis. Is that basically what you were asking?
Yeah.
And the same thing, you look on the y-axis and it tells you the same information, but for a different formula to take that complicated spatial pattern. And now basically you can imagine, and the way it works is principal coordinate 1 is the way to best represent the distances that everything are from each other. That's why it's PC1. It is the mathematical formula that separates out the samples as best as possible. And principal coordinate 2 is the next best. So in the text, they then even say somewhere, what percentage of the total, oh well, I probably didn't include it. They say, I think principal coordinate 1 represents 17% of the total distance space.
Yeah, I think it's
Okay.
the percentages correspond on the axis.
Oh yeah, right, they show it. So PC3 represents 7.5%, PC4 you're getting less and less of the total distances represented as you go for, yeah, so 16.5 and then 10%. So we can go into principal coordinate. I wouldn't worry about all the details here. It is a way of representing in 2 dimensions the distances that communities are from each other in this unifrac distance space. That's probably the most important thing. And then they show some of this sort of jackknifing results. They go through all of their questions. How does culturing affect things? They make, I don't want to go into all the text about this, but they basically, try to scan through all of their results to compare within a particular samples, cultured versus uncultured organisms. They do the same thing with organisms that are widespread across geography. Again, they say, they change the question a little bit. That's what I was saying, how cosmopolitan does geography play a role? They go, and again, I want to get to the other paper, so I'm not going to go into all the details of all their conclusions, but are marine ice, sediment, and seawater 3 distinct homogeneous habitats? They're basically looking for whether or not they all group more similar to each other than they are to anything else. And they don't completely, but they do a little bit. And then they basically say, the point of this paper is not any of the biological inferences that they made. This is before we had large samples of anything. The point of this paper is a method. And they say that, UniFrac is great. It can integrate sequence data from lots of environments, do large-scale comparisons, and really importantly, despite there being differences in data collection techniques between samples. Although in the other paper, I think there's a little skepticism of this. They then say, oh, we know that we're not counting multiple, if we have differences in relative abundance, but that's not currently represented in UniFrac. They know that they're working on, when you write this paper, you always put in the end of your paper what you're basically working on now so that you can refer back to it about how brilliant you are for solving this horrible problem that was pointed out in your own paper. And then, the conclusions of their paper are not really about any individual environment, but are about the future potential of analyzing 16S data and environmental information using tools like this. And they were right on this. I mean, this is, there's probably 25 papers a week now where people are doing something like this for different microbial communities. And almost all of them using UniFrac as part of their workflow. And then the Hughes paper is, I love this paper. I think it's just completely, I refer back to it all the time. And it really does lay out, I think, a lot of what you need to do in doing other types of ecological comparisons about communities. The introduction is basically saying nobody's working on microbes at the time when they were doing this. Nobody's doing sort of the ecological metrics for microbes. They point out there are only like 2 papers where people have done this for, using this type of sequence data. And they basically lay out in here ways to compare different approaches for analyzing the diversity of samples. this is kind of, I don't know why they put this as the header of one of their sections. Maybe a reviewer made them put it, but they clearly don't think microbes are too diverse to count. The whole paper is about how to count microbes. So, and so then they go through, 3 basic approaches to doing this. An accumulation curve, and they talk about, what an accumulation curve is. They show you the accumulation curves in figure 1 for these 5 different communities, and this is an accumulation curve. So an accumulation curve is basically a normalized way of comparing how you've the diversity of a community by taking a sample of the community and asking, as I sample the community, what fraction of the total diversity in that sample do I see as I sample it? So if you sample 10% of the community, what proportion of the total OTUs do you observe? So if you observe 10% of the OTUs, that basically means that you've never seen one OTU twice. If you sample 10% of the community and you observe 70% of the OTUs, that basically is telling you that there are some OTUs that are really abundant, that you're hitting, a lot of the community with a lot of the number of OTUs when you've just sampled a couple of things from the community.
What is it? But I don't understand how they determine the proportion of individuals sampled when they don't know the diversity of the community.
They don't. This is within a data set.
Oh, okay.
So this is not trying to estimate the diversity. This is taking a data set that you have. Let's say you sampled and you found 50 OTUs or 50 species. And it's just a way of showing in a normalized way what the shape of the accumulation curve is. Accumulation being, as you collect samples, how many new species do you see as you collect samples? That's why it's called an accumulation curve. And it's nice to do this sort of normalization, so now you're sort of able to compare, even if you have only 20 individuals for one community and 200 million for another community, you've sort of normalized them by this type of graph. And that's why a lot of people use it, to just get a general idea of the shape of the curve of diversity in a community. And then they talk about how some people have used accumulation curves, and they think probably not well, to say that microbial diversity cannot be estimated or cannot be measured, because even though it's nice to normalize across communities, it also doesn't show you the richness of the data when you compress it this way. So there's another approach, which they go into also, which is this rank abundance curve, which is another way of looking at the relative abundance of different taxa in your community, probably a much more useful way, because you can have interesting differences in rank abundance and get similar accumulation curves. And here is comparing, I forget what it is, tropical moths versus soil bacteria. It's reversed, it must be. Yeah, I think this graph is reversed. They're just basically showing you simple ecological ways to compare communities here first. These are not their favorite ways. But they do say that you can probably use the different methods that have been used for plants and animals to study microbes. I don't think they anticipated how much sequencing people were going to do from some communities. And they say, but we're never going to sample a community exhaustively for microbes. It's just not going to happen. So what we need are estimates of the diversity rather than measures of the diversity. So we need ways to statistically look at the shapes of these curves, or this curve, and estimate what the true diversity is, or look at the data maybe in a different way. And so then they go through tools for doing these estimates. And so they say, there are lots of tools available for making estimates for macroorganisms, and they discuss 4 that could be used for studies of microbes. And all 4 in different ways have been used for microbial diversity studies. So rarefaction is similar in concept to the accumulation curves, but you do things a little bit differently. Anybody know or want to try and explain what rarefaction is? Yeah, me too for a long time.
Like the average, the sort of rank abundance curve. Like to me, it sounded like they basically said, well, the data from the rank abundance curve is sort of stuff we get from the environment. It's not pretty and nice, and if we average the things out, we can fit something to the curve and get an estimation of when it will sort of flatten.
Yeah, that's exactly right. So you take, I mean, there are many reasons why you would want to do it, but you basically take that accumulation curve, and rather than just plotting one iteration through the accumulation curve, you do statistical sampling of the data, randomize the data in various ways, and basically analyze the variance that you see in the randomizations of an accumulation curve. It's also, like accumulation curves, can be very useful for comparing sites that have been sampled differently, but now you get some statistics to go with it rather than just, so right, if you look at the accumulation curve, basically what you're going to try and do is assign statistical values to some of the shapes of this curve and some of the parameters that you see, and use that to understand, you know, if this is the shape, but I've only sampled 10 individuals, maybe there's a massive amount of error around that, and it just doesn't mean anything to have that shape compared to some other shape. I'm running out of battery power. I may have to plug in my computer. And they don't really show this yet, but then they go into richness estimators. Richness estimators. Richness is what? What type of diversity? What? It's the total number of species.
So it doesn't take abundance into account.
It doesn't include anything about abundance. It's how many species do you see in a particular environment. And they talk about in this paper that they're going to focus a lot on richness, but it's not the only thing you would want to measure about a community. And then they say, there are these many classes of richness estimators, and they go into 3 of them. And these are sort of the 3 other things that you would do with microbial communities. So one is really simple. It's curve extrapolation. So you look at the shape of your curve. We talked about this before, and you try and basically determine whether or not you're asymptoting and where you might asymptote with the curve. So you do curve extrapolation. I don't think they like those very much. They don't spend a lot of time on it, largely because they feel like for microbial communities, you're mostly in the part of the curve where none of them are approaching an asymptote. I mean, if the curve was like this, you could use a curve extrapolator and say, yeah, you know, we're not going much above 11 or something like that. But, you know, they certainly didn't feel like this was something you could use, although that, I've mentioned this before, that is now changing because you can actually start to asymptote in many microbial communities now. So people, I think, will return to curve extrapolation for many purposes. And then they say there are these 2 alternative approaches, parametric estimation. Anybody want to try to explain what parametric estimation is? What is a parameter? No one? So parametric estimators versus non-parametric, the difference between them is parametric estimators try and use a parameter about the community, a model basically, as part of their way of estimating the diversity. And the non-parametrics basically try to avoid using some specific parameter or model, although there's gray areas between both of them. They talk about a diversity of parametric methods used to estimate species abundances and how those parametric methods, and I mentioned this before, have a model, for example, about the shape of the species rank abundance curve behind them. And then if you have a good model of the rank abundance curve, you can convert your observed rank abundance curve to an estimate of, what's at the tail, basically. And they sort of explain it here. They estimate the number of unobserved species by fitting sample data to models of species relative abundances. And then they say, we don't like those. This is the standard met, right? I mean, they're going through the things they don't like first. You need data on species relative abundances, which they were at the time skeptical that people would get for microbial communities. We're now starting to get things that might be useful measures of relative abundance, so that may not be as big of a problem. You have to make an assumption about the distribution in a community, and we know very little about what those shapes look like for microbial communities, so they weren't comfortable, and I agree with them on this, about what assumption we would make in order to have a model that would then fit the data to the model. And then they say, finally, even if one of those models is a good approximation, parametric estimators need lots of data to work, because you're basically, if you have a wide species rank abundance curve, you're trying to do a lot at the tail end of this curve and at, closely fitting data to the model. And they're worried because the largest microbial data sets only include 100 individuals. Now, as we talked about, for $1,000, you can get 20 million sequences now. So I don't think that this is a problem anymore for the parametric methods. They actually seem like they're much better than they used to be because we're able to get much more data. And then the non-parametrics are just methods that don't at least have an explicit model behind them. Sometimes when you dig into them, you can actually figure out that they need a model behind them sometimes. But the ones that they talk about here, this is only a subsample of the non-parametric estimators that you could use, are these ones that are modified from this mark release recapture statistics. And so in ecological studies, this is very common, or actually not just ecology, but human demography and studies of Facebook and other, people use mark release recapture concept in all sorts of different ways. And I think I described this before, but it's very simple. If you want to estimate the number of butterflies in a field site, you go to the field site and you collect, let's say, 100 butterflies, and you literally mark them with a Sharpie, for example. I've done this. And then you release them, and you come back the next day, and you collect 100 butterflies, and you ask, how many of the marked ones do I see? And if you recover lots of marked ones, that says that your population is small. And if you don't recover lots of them, it says your population is big. Now, that approach doesn't make a model assumption about the shape of the sort of relative of the pattern of the butterflies, but it makes all sorts of assumptions about that they're not moving between communities, that you're sampling them at a time when all of them can fly, and so there are lots of assumptions in these. There's no mathematical assumption there about the butterfly community per se. And people have adapted this approach where instead of marking individual organisms, your mark is whether or not a sequence goes into an OTU that you have observed or not. So if you sample a community 100 times, you ask basically, for the first sequence, obviously it's going to be in a unique OTU. But for the second sequence, is it in an OTU that I've observed before, or is it in a new OTU? And for the third sequence, is it in one that I've observed before or in another OTU? And that's the two methods, the Chow versus ACE method that they go into in this paper, have different approaches to making estimates of the community from a mark release recapture type of math. And the Chow does this in one way, and the ACE does it another. They go through them in detail. The Chow is really simple. This is the original Chow diversity index. S observed is the number of observed species. So if you sequence 100 ribosomal RNAs, how many OTUs do you see? And then this formula is the number of singletons. That is, now this was not derived for ribosomal RNA. This was derived for mark release recapture experiments, but we're going to apply it to ribosomal RNA. For how many times do I observe an OTU once? That is the number of the 100 that I look at. How many OTUs are observed only a single time in that data? And how many OTUs are observed twice? So this is another important thing. For mark release recapture, you could capture the same butterfly 3 or 4 times. That tells you actually extra information over capturing it just once. And so this, you can take this and then make an estimate of the total diversity from this formula. And then the ACE diversity index is a little more complicated. Basically, what it's trying to do, so Chow only takes into account the number of times you see something once or the number of times you see something twice. That's it. That's the entire amount of information in this diversity index. And the ACE index says, let's be a little more complicated. Let's take into account rare things in the sample, abundant things in the sample, and have a much more elaborate calculation of the sort of coverage of a sample that you get from this approach. I wouldn't worry about the exact per se math here. The main difference is that Chow, again, only looks at these singletons and doubletons, and the ACE looks at much more of the data. Each of them have a way of calculating the variance in your observation, a direct sort of mathematical way. And then the rest of the paper is about evaluating these metrics. So they talk about whether or not there's going to be bias in the approaches. They define, you should look at its very useful description of some of the statistical terms that people use. So they go through and say what bias is. Bias is the difference between the expected value of the estimator and the true value, basically. Precision is the variation of the estimates between each other. So if you have you make many different estimates, do you always get the same estimate, or do you get different estimates? And then they talk about the formulas to calculate this, and they go through really for one data set, although they say for 4. A human mouth and gut sample they focus on most. They tell you a little bit about the data set, and then this is the key part of the paper. So here is the observed versus estimated richness for this data set given the Chow diversity estimate and the ACE diversity estimate. So here's the plot. Here's the observed. So how many clones did you sample? How many OTUs do you actually see in the sample? And then, really interesting, they plot what happens to the estimate. So let's say you take only 50 of your clones, and now you run the Chow diversity index on that 50 of your clones. What does it estimate the total number of OTUs are in your sample? So you can see there were 20 OTUs. The Chow diversity estimator, if you had 50 of them, will estimate that there are really 40 species, 40 OTUs. The ACE will estimate a little bit different. Oh, sorry, ACE.
Yeah.
ACE is the number of OTUs per given, or the accumulation versus average. Okay, the ACE is the triangles, and the Chow is this. Why is the actual observed higher than the estimate? That's because the methods are somewhat biased, probably. I'm gonna have to, this doesn't look right. Let's ignore the actual observed data, because I think there's something a little strange there. Basically, what this shows is how your Chow diversity estimate changes as you get more and more data. And it's not very stable, right? That's unusual that you're estimating the diversity in your sample, but the estimate of diversity goes up as you get more sample, which is really not, that means your estimator is not very good, right? And the same thing happens with the ACE index. So these indices are not particularly good for, at least for low numbers of samples in the data set. That is what they were hinting at. You need lots of data for some of these to get good estimates. They then go through just for the Chow diversity index for two different types of samples, mouth and gut, and they do the statistics here. They're trying to say, if I add the variance in the Chow estimator, can I see whether or not one of these communities has higher diversity than the other community by looking at whether or not they have significant differences, and they basically does not seem like there are big differences between these two communities. They then look at the precision. In essence, as you get more and more clones, does your estimator get closer and closer to each other, multiple different estimators from that sort of same number of samples? So the method for the mouth and the gut does get better and better as you get more and more data, in essence. You want to have the range in your 95% confidence interval go down as you get more and more data. Yeah.
They actually fixed the figure 3. The data is wrong.
Oh, you're kidding.
So the circle ones, the light circle ones, that's supposed to be for Chow.
Oh, thank God. I do.
And the triangles is supposed to be
Okay, good. So there's a correction to this one?
Yeah, I remember them being at the bottom.
Yeah, okay. Because as I was just trying to say, it didn't make any sense. All right.
Oh, errata.
I'll have to go add that to my slides. This is then a rarefaction curve showing, in essence, it's an accumulation curve, but now with statistical error bars that can show that, what appear to be real differences between the mouth and the gut. Here, they do a little bit on the aquatic ecosystems. They do a little bit on Scottish soil systems. And then they go through a few conclusions about what this means. And I think because they're going to kick us out of here, I will leave it there. But basically, I think that this paper is really good at laying out the general issues of different types of estimators, about parametric versus non-parametric, about different ways to go from real data or observed data. And that's basically everybody since that paper is just adding new methods onto mostly what's described in here, new ways to do it.